ಈಗ ಕೇಳ್ತಾ ಉಂಟಲ್ಲ. ನಾನು ಎಲ್ಲಿ ತನಕ ಮಾತಾಡಿದ್ದು ಕೇಳ್ತು ನಿಮ್ಗೆ? ಎಲ್ಲಿ ತನಕ ಮಾತಾಡಿದ್ದು ಕೇಳ್ತು? ನಾನು ಏನೋ ಹೇಳ್ತಾ ಇದ್ದೆ. ಪ್ರೋಟೋಪ್ಲಾಸಮ್ ಎಲ್ಲ ಕೇಳಿದ್ಯಾ? ಹಾ, ಲಾಸ್ಟ್ ಬ್ರೀಫ್ ಒಂದೇನಾ? ಹಾ, ATP ಸಿಂಥಸಿಸ್ ಸ್ಟಾರ್ಟ್ ಆಗಬೇಕು ಅಂದ್ರೆ ಎನರ್ಜಿ ATP ಇರುತ್ತೆ. ಆ ATP ಸ್ವಲ್ಪ ಇರುತ್ತೆ ಆಯ್ತಾ, ಅದು ತುಂಬಾ ಹೊತ್ತಿಗೆಲ್ಲ ಎಂಡ್ ಆಗಲ್ಲ. ಅದು ಇಟ್ ಕ್ಯಾನ್ ಓನ್ಲಿ ಗೋ ಫಾರ್ ಲೈಕ್ ಓನ್ಲಿ ಫ್ಯೂ ಮಿನಿಟ್ಸ್. ನಾವ್ ಇಟ್ಸ್ ಡನ್. ಓಕೆ, ಸರಿ, ಅದು ಯೂಟ್ಯೂಬ್ ರನ್ ಮಾಡ್ತಾ ಇದ್ದೆ. ಸೊ ಓಕೆ ದೆನ್, ಸೊ ಐ ವಿಲ್ ಸ್ಟಾರ್ಟ್ ರೆಕಾರ್ಡಿಂಗ್. Good evening everyone who are in YouTube also and who are in here is also. Okay, so 53 people are here, and I will inform last one time also. This week, that means this Sunday, you guys have the chapter test on Animal Kingdom. Okay, remember? Hi Ashwin. So I will put a message in the in YouTube also. This, one second. This week we have on Sunday. Okay, so be prepared. Good evening. Okay, so everyone be prepared. So same pattern, 40 questions will be there. Every correct answer will get plus 4 marks. Every wrong answer will lose 1 marks. Okay, minus 1. And if you won't attend or if you're not attend any questions, then there is no gain or lose. Okay, so total will be 40 minutes and 40 questions. Okay, everything is multiple choice only. And who are in WhatsApp, you know how to attend the test and everything. Who are in the Telegram, that means who are watching from the YouTube, I will share the question paper in YouTube, a Telegram group, and I will also put a message in there. Who want to attend, you can attend the test and you can answer in the link, what's the Google form link, which I send it through there only. Okay, so we'll see. If you have any doubt regarding that, once I put a message in the Telegram group, you can reply to that. Okay. Good evening, Gopika. Chemistry and physics test is it may happen. Probably then you guys have three subject exams then. Okay. Then like you people may have 3 subject exam, physics, chemistry and biology. So it's great then. So from writing single subject, you will come to a stage where you can write 3 subject, like 3 chapter test. So prepare well then. So this actually matters after 1 year, almost 1 year in the 11th. Okay guys, then we can start the class then. We thought of completing this yesterday only, but because of my absence, it's not completed yet. We will try to complete it today. Okay, so I added some more slides to it, so make it in a correct time. So we are discussing about eukaryotic cell. In yesterday, I think you discussed about the cytoskeleton, cilia, flagella, and all the things. I think you guys know about it better, right? We will start the questions then. First, CFU came simple. Okay, the human sperm flagella have, they are asking, the human sperm flagella will have 9+1 system, 9+2 system, 8+2 system, 7+2 system. Okay, good evening Darshan. Okay, I'm launching the poll. You can answer in the poll. So yes, I'm launching it. Okay, simple question, very simple answer, like you can choose it in a second, right? Good evening Venkat. Change your name, dude. Whenever I thought of calling your name, I will like struck 1 second. Okay, then I will go to the second name, then I will call you. Okay, Ashwin answered quickly. Good. Okay, some of you got confused, but most of you are giving correct answers. Gopika. Others who are in YouTube, can you guys answer this? You have 10 seconds left. I'm closing the this poll within 10 seconds. Okay, within that I'm expecting more responses. Okay, then I will close it. Right. I'm sharing the results. 12% chosen option A, 63% chosen option B, 18% chosen option C, and 17% chosen option D. Okay, in YouTube, everyone chosen option D only, right? Human sperm flagella. So when we come to the human sperm flagella, so what's the meaning of human sperm? Is it means it's a eukaryote or a prokaryote? What do you guys think? What do you guys think? Is it a eukaryote or prokaryote? Human sperm. Exactly. It's a gamete, but it's a eukaryotic gamete, right? Exactly. It's a eukaryotic gamete. So if it's a prokaryotic gamete, then it's a single flagella without any differentiation, right? A single flagellin protein will cover this, right? Single flagellin protein will do something like this. But in human sperm, it's a eukaryotic structure. It's actually a circular with the central two uh, things with these surrounding 9 things, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, like this, right? With every spokes and all those things which is present. So something like this, you can write it, right? So it's a 9+2 system. So 9 surrounding or peripheral tubes and 2 central tubes, right? This is the system what we are following. So you can combine it to form 11, but we call it in 9+2 only because that is how it is arranged, right? So the option is 2. This question is very important. It may be asked in different forms, right? Human sperm flagella have dash system, they may ask in CBSE, okay? Because you have to remember this. This is easy, right? 9 pairs and 1 pair in 9. 9. You can call it like 1 second. We will go back here only. You can see, right? So 1 sec. Exactly. So here you can see the bundles, right? A peripheral microtubule doublets. So instead of using the term, you can use the term doublet. Okay, both are related. Doublet. The only difference why we are saying why we are using the term doublet, not the term pair, because both of them are not same. Okay, both of the microtubule circle are there, right? It's not same. It's slightly different in bondings. Okay, that's why we are using the term doublets. Okay. That's why. Yes. Look, you can see here, right? They use the different colors. Why they use the different colors? Because little bit they are changed. One is a alpha tubule, another is the beta tubule. Okay, so that's the difference. Leave it. That's not in the syllabus. That's secondary. Okay, so that is what related to here. 9+2 system. You can say correct for 9 pairs. You can say 9 pair in the this one pair in the middle. Okay, actually, oxygen. Exactly. So when you take bacteria or any prokaryote, their flagella don't have this system. They have a single flagellin protein. Okay, single flagellin protein. Correct. Right. Okay. So we will move on to the second question. This is also easy. What are flagella and cilia of eukaryotic cell is made up of? Okay, they're asking what are flagella and cilia of eukaryotic cells are made up of. Okay. Option E is tubulin, option B is flagellin, option C is lamina, option D is keratin. Okay, I'm launching the poll so you can answer in quickly. This is also easy question, right? So everyone can answer properly. Don't confuse. So every word have certain meanings or importance. Yeah. In YouTube, I got answer quickly. Yes, done. Awesome, guys. Awesome. Okay. Yeah, you gave answer within like how many? 10 seconds. That's really awesome. So that should be your most, right? This is question asked. Really? Oh, sorry. I didn't. I thought that this question is not asked yesterday. But I changed this one options. I changed the options. Okay. Then there is no need to answer. Okay, I'm ending the poll. So many people give the correct answers and still some people have the confusions. No issue. I will solve it one more time. Okay. They are asking about flagella and cilia. Exactly. Okay. 1 second. They're asking about flagella and cilia. Tubulin is the material which actually make flagella and cilia is possible in the eukaryote. Okay, in the eukaryote. If you go for a flagellin, yeah, flagellin also make flagella, but this is in the prokaryote. Okay, prokaryote, and that's also related to the flagella only. Why flagella only? Because cilia is not present in the prokaryote, if you remember, right? Flagella is not present in the eukaryote, and cilia is not present in the prokaryotes, and only flagella is in the prokaryote, but that's also made up of the flagellin protein. Okay, lamina is just a membrane. Keratin, you know where keratin is present. Okay, if I ask you a question something like this, what are which one you can choose? We can choose. Um, Pillai. Okay, pillai of bacteria is made up of. Pillai of bacteria is made up of. If I ask you a question, if what is pillai of bacteria is made up of, then what will be your answer? What will be your answer? What is your answer then? So exactly, I got one answer, two answers, three answers, so many answers. Exactly. It is made up of the pillin or pillin, right? Pillin. I'm not sure about the spelling. Sorry. Okay. So, pillin. So, pilli of bacteria, or sex pilli of the bacteria is made up of the pillin, right? Okay. So, you have to remember that one. So, for this question, eukaryote, the correct answer is tubulin. That's why I mentioned the eukaryote name. Yeah. If I know that, I could have changed the question. Okay, no problem. So today we are going to the next one. So that's about the nucleus. Okay, nucleus is the center of the cell. Center of cell. It is actually first observed by the Robert Brown. Okay, he observed the nucleus, a dark part which is present in the cell. Okay, center of the cell does not mean that nucleus will be present always in the center of the cell. It simply means that it's controlling the cell. Okay, it's exactly like the capital of the state, right? The capital of states or a nation should not be present in the center of the state, right? We will say that capital is the center, right? Capital is the center, or we simply call it a city center. But that does not mean that that particular will be present exactly in the center. It may be present in the outward also, right? In Karnataka, the state capital is Bangalore, which is present in a tip in the southern part, right? So in Maharashtra, it is present in the coastal side, another one sides, right? And if you take Delhi also, it's present in the almost upper region, neck region of the India, right? Oh, centrosome not over. Okay. We'll complete nucleus. We'll go back to that. Okay, no issue. Yep. So that's what this is called the center of the cell. Okay, nucleus is the center of the cell, and it is actually bounded by nuclear membrane. Bounded by nuclear membrane. And what else? What it contains? Contain DNA. Contain DNA, or in simple terms, you can call it as like genetic material. Okay, so both of these words will how can I say, like, mean same in eukaryotes, right? If I'm talking about some virus, then if I mention genetic material, then I have to mention you guys like, is it DNA or RNA? But if I mention the word genetic material in the eukaryote or prokaryote, it directly means the DNA only, right? Exactly. It contain the DNA genetic material plus nucleoplasm. Nucleoplasm, correct? Nucleoplasm is the Hachitana, you raised your hand. So it's a how can I say? So liquid which is present in the within the nuclear membrane, right? And also the nucleolus. So these are the things normally present inside to that. Okay, which are the things which is present inside to that. Okay, so when you consider egg as your cell, okay, don't consider yellow as a nucleus. Okay, nucleus is tiny only, still is tiny only. It is present inside that yellow region. Okay, all those yellow is a kind of a protein which gives that particular color, and white is also protein only. Okay, both of them protein only, both of them energy materials. Okay, so nucleus contains the DNA or genetic material and nucleoplasm, and the nucleolus. Okay, What left then? So it's the main part. It's controlling all things of this cell. Or instead of saying the things, I can say activity. Right, all the activity is controlling by that particular cell, exactly like how our center is controlling all the state matters, right? Today's gram panchayati result is going on in Karnataka, right? How the gram panchayati will work? Gram panchayati will it's a center of the system and it's doing the works, right? What should do and how the things have to go and everything, right? That's what the nucleus is doing, right? So second things, if you go inside to that, I will mention it one more time. Okay. So nucleus only present in eukaryotic cell, only present in eukaryotic cell. Why? Because in prokaryote you can't find the nucleus, right? Eukaryotic cell. Eukaryotic cell. Then what about prokaryotes? If I ask you about prokaryotes contain nucleoid, right? Nucleoid means a naked DNA. They don't have the nucleus. Naked DNA, they don't have the nucleus. So it's simply in a box. Okay. It will not control activities of mitochondria. It will not control the activities of the bacteria, but still some of the products, gene products, mRNA or proteins are necessary for the bacterial membrane and the production of their enzymes. Okay, even though it's not at all controlling the things, that means how it will activate or how it will divide, but it has some function over it, some control over it. Okay, that's why if you remove this one thing, if you remove the nucleus, other organelles normally won't survive. Okay, the major reason is in directly or indirectly the nucleus supporting all the things. Okay, so that's what, exactly. So we'll see the interface nucleus contain. They are saying we got with another one question. Then what is interface then? Okay, when a cell is dividing, we we divide this into two stages. Okay, first stage is the interface where actually it's getting ready for the division, and second one is the metaphase. Okay, second is the metaphase. So here actually this division is happening. Okay, so interface is like getting matured state. Okay, what is the genetic material in prokaryotes? DNA is the genetic material in prokaryotes, but in all prokaryotes, all eukaryotes, the genetic material is DNA only, except in a virus. In virus only RNA. Okay, so this interface nucleus, why we consider this one then? Okay, because when it's going through the dividing state, the structures are different. Some structure will be dissolved. Dissolved means disintegrated. Okay, some structures may be builded. Okay, but in interface is a common state. If you take any cell of your body and if you observe, most of the cells are in interface state. That means in a growing stage. Okay, that's what they are mentioning. The interface nucleus contain, exactly, Manvita, right? You can also call it as resting phase. One time you called it as a resting phase. Okay, we don't, it's like something like you guys. Okay, exactly. Okay, your father and mother, everyone think that you guys are sleeping. Okay, but you guys only know that you are reading. Okay, even though it may not, that's secondary, right? So whenever they call you like, why are you sleeping, you will tell by opening the book, I am reading, I am not sleeping. Exactly. So all of us thought that it's a resting phase. Okay, we thought that it's a resting phase, but then we realized that no, it's not a resting phase. You guys are true. Okay, it's actually doing a lot of work. Exactly, it's actually getting prepared for the metaphase. Okay, getting prepared for the sorry, why did I wrote metaphase? Not metaphase, mitotic phase. Okay, mitotic phase. Sorry, mitotic phase. That means a dividing phase. Okay. So now we can say it's one of the highly reactive phase interface, but you can think like separate. We will see that in the third chapter. Okay, no, metabolically active, exactly, Manoj. Okay, this interface nucleus contains four different groups. One is it have a nuclear envelope, that means a covering. Okay, second one is the nuclear matrix. Matrix means a fluid, that's called nucleoplasm. And the third one is the chromatin, or you can say a form of DNA. Okay, DNA a formation. We will see what is the chromatin, all the things. Correct, Mankad. And nucleolus, another one dark structure which is present inside to that. Okay, if you observe this, you will get to know, right? There is a nuclear membrane. It's actually strongly adhered to this rough endoplasmic reticulum. Okay, then you can find a chromatin. Chromatin means the DNA particles which are present here. And in center, you can also find a dark structure. It looks like center within a center, right? It also looks like nucleus of the nucleus. So we gave the name nucleolus. Okay. Some people think something like this also. I think at least one of you probably thought like this. When you boil your egg, okay, boil your this regular chicken egg, everything will solidify, right? So then what happens? If you make an exact cut in the middle, in the center, you will find a yellow color thing. Within that yellow color thing, you will again find some darker color thing. Okay, a point, something like that. Within that yellow color oak, you can again find another one thing. You can think that probably the yellow color is the nucleus. Inside that again one dot is there. That is the nucleolus. Okay, it's not true. Okay, I told you, right? The yellow color is not the nucleus. Then that is not the nucleolus. Okay, so probably some of you probably thought like that. That's why I thought of clearing that out. Exactly. Okay, so never think in that manner. This is the endoplasmic reticulum, and this is the nuclear membrane inside that chromatin and the nucleolus. Okay, so we will move on then. We will move on to the nuclear envelope. Actually, before to that, when I'm going to the nuclear envelope, so I got to know, right, we haven't completed the centrosome properly. Then I will finish this, then I will go back to there. Okay, we will take a small break here then. Okay. So centrosome, probably a little bit, right? What are centrosomes? These are organelles. Yeah, these are organelles. These are like pipe-like organelles. Okay. How can I say? So these centrosomes are centrioles, both are alike only. Okay. What they are made up of? They are also made up of the microtubules only. Okay. They are also made up of microtubules only. How is the arrangement then? They arranged in a 9 triplet fashion. Okay, 9 triplet fashion. As you can see here, I will just name it one by one. 1, 2, 3, 4, 5, 6, 7, 8, and 9. As you can see. Okay. Remember, it is not 9 plus 2 system, where you can find 9 outer and 2 inner, right? But here it is 9 triplet system. Okay, 9 triplet system. Right. What's the meaning of 9 triplet? In a single branch, you can find the 3 microtubules. Okay, but center there is nothing is present. That means center is hollow. Okay, center is hollow. You can say. Okay. The 1 second, guys. Yeah, nothing. Yeah, center is hollow. Okay. So this is what this 9 triplet thing, you call it as the centriole. Okay, centriole. Then what is centrosome then? Okay, what happened during cell division? I'm writing it down because you don't get confused. During cell division, what happens? Centrioles, 2 centriole exactly, arrange 2 centrioles arrange in crisscross manner, and that is called centrosome. Okay, so simple then, right? So as you can see here, a centriole is structured like this. If the same centriole, if I take the same centriole, if I arrange it somewhat like this, one centriole like this, okay, and if I arrange other one like this, okay, then you can call this as the centrosome. Sir, do centrioles duplicate themselves? Every organelles will duplicate because of this thing. Centrioles are made up of the proteins, right? The proteins will be created during the interface. Those interface created proteins will initiate the process. Okay, it's all controlling the from the DNA only. Like whenever they are going for the cell division state, then only they will go to duplication. Okay, otherwise they won't. I will just rub up some of the things. Okay, you can find something like this. Okay, this is, you can call it as the centrosome then. Okay, what's the function of centrosome then? That's very important, right? Okay, we heard what is centrosome, centriole is. Centriole do not have any function. Okay, don't think like that. Centriole normally present in there, normally controls some microtubules. Centrosome is very important. The function is production of microtubule, or you can call it as exactly correct Manvita, spindle, correct Kirtana, spindle fibers, or asters during cell division. Okay, when cell division is happening, then they will produce the microtubules. You can also call it as like spindle fibers, also call it as like asters. Okay, sorry for my handwriting. Okay, it's not at all good, so I can't. So leave that one. Okay, we will concentrate on the things. You write your notes. Okay, so microtubules, spindle fibers, and asters. But when you are using the aster term, you should be very careful, because we use the term asters only in animal cell. Only in animal cell. What is the reason for then this? What is the reason for using the animal cells only? That I will come to. Okay, this centrosome is only present in animal cell, only present in the animal cell. Okay, remember, not only the chloroplast only present in the plant cell, right? Exactly opposite to that, the centrosomes only present in animal cell, not in the plant cell. Okay, only in the animal cell. Okay, that starts our another one doubt, right? What is that doubt? Can anybody answer me? What is that doubt? If I told you that animal centrosomes only present in animal cell, what doubt will arise for you guys? Anything or nothing? Fungi protist. Correct, right. When I told you it's only present in animal, what about the fungi, protist, or plants? Right. Which product? How cells get divided in plants then? It do not have the centrosomes then. Exactly. Remember this thing. Okay, exactly. Remember, the centrosomes are not at all necessary for production of the spindle fibers. Okay, if you take a particular cell, and if you are going for like production of the spindle during the cell division, centrosomes are not at all necessary. Okay, then how this will help in animal cell? In animal cells, presence of centrosome help in orientation of the spindle fibers more better. Okay, it's like having an additional feature, right? When people are taking cars, there will be different models, right? One will be the base model, one will be the middle, like 2-3 models, and last one will be the highest top end model, right? I think that I took the base model, and Tanmay took the top end model. Okay, and Venkat took the middle or something. Okay, then what will be the difference? Does the car is different? No. Does the car move? Exactly, it will move. Okay, does the car have seat? Yes, it do have the seats. Then what will be the variations? I have to raise the window by turning that lever. Okay, but Venkat and Tanmay can do it by pressing the button, because they have the power windows, right? I may have the regular this ones. What is that? Steering. I have to rotate it like 10 times to turn it back. But Tanmay and Venkat have power steering probably. If they just turn it like this, it will take a rotation, right? That means it's an upgrade, right? It is not a necessary thing. So still I am also going, and both of them are also going somewhere, right? Then what will be the difference happen? All of us are going only, but the difference is what we are enjoying. They are enjoying some more facilities, right? Exactly. Having centrosome is having more facilities, make activities fast and easy. Exactly. Then There's one thing, it's not in the syllabus. I just mentioned it. Okay, don't get it too seriously because not in the syllabus. So that's called MTOC. Okay, MTOC. MTOC means microtubule organizing center. Organizing center. Microtubule organizing centers. Okay, so this microtubule organizing center is the one which actually producing or organizing the spindle fiber. Okay, so what's the advantage then? What's the advantage then? I will tell you. Okay, so think that this is the, one second, I will change the color. Think that this is the animal cell. Okay, and another one I will write down. This is the plant cell. Okay, during cell division, what happens? What is present in the MTOC? MTOC is a group of enzymes. Okay, group of enzymes which actually taking care of the it's like some proteins. I can't say all enzymes. Okay, there are some proteins are there, certain enzymes are there. They help in joining together of the microtubule particles. Okay, so it's like a little bit complex. And remember, here centrosomes are there. There is no centrosomes, only MTOC are here. Okay, then how the microtubule formation will happen? In this one, microtubule will happen in all the ways. Okay, in 360-degree microtubule formation will happen because of the presence of the centrosome. Okay, but in animal cell this only happen in one way. Okay, happen in the one way. Okay, I will mention the names also. This is this is the plant cell, this is the animal cell. Okay, look how it is looking for you. This is like centrosome, everywhere it's radiating, right? It looks like star, right? It looks like the star. We already know the name of the star, like we call it as the aster. That's why we call centrosome in animals asters. Okay, where only in the animals, because it's giving rise to the microtubule in all directions. Okay, so we call it as the asters, and we call it as these particular things into aster rays. Okay, we call it as the aster rays. So that means radiating a light kind of the things which is started from the aster or started from the star. Okay, but plant don't have all those things. It only radiate in a one particular way. So instead of calling it aster, we call it as like spindle fiber or microtubule. Okay, that's the difference. They may ask you, like if they mention the term aster or aster ray, remember they are talking about the animals, not about the plant cells. Okay, these are the things related to centrosomes and the centriole. Drag the center, but how it's specific? Correct. Akshay is asking, like in animals, like the centrosomes will pull back the things, so that will happen, right? What about the plant? That's not at all true, actually. Okay, there is no pulling force will happen. How can I say? It's like a simple term. I will put it like this. Okay, think our microtubule is like this. Okay, it's made up of several tubulin protein, and it formed like this. What happens in here then? What happens in here? So think that this is the centrosome. Okay, this is the centrosome. Here its chromosome is attached. Okay, what is happening is, in this particular region, when after the metaphase, when they want to pull the chromosome back, these things are getting depolarized. Sorry, these are getting removed from this end. Okay, when these are getting removed from this end, this new end will come and join to this particular centrosome, and it's going in this manner. It's going in this manner. Okay, that's why, like one side polymerization will be happening. That means new things will be adding. Another thing, depolymerization will be happening. The tubulin molecules removal will be happening. Okay, exactly, Mamita. Microtubule length is getting reduced by the depolymerization process. There is no pulling kind of the thing. Okay, pulling. I will keep it in that way only. Okay, I'm not going in depth to that because it's like too much a big chapter. Okay, this particular centrosome only. That's why. This comes again in cell cycle. Little bit it again comes in this cell cycle and division. Correct. Okay, you got it, Venkat. Okay, so this is how it happens. Okay. Yeah, we can move on, right? So anything left, guys? If you have any doubt again related to the centrosome. Okay, we will move on to the nuclear membrane then. Okay. Nuclear membrane then. So it is having like two layers. Remember, it having the two layers. Okay. The outer layer, what you can see here, outer layer is actually continuous with endoplasmic reticulum. With endoplasmic reticulum, normally rough endoplasmic reticulum. That means if I write the layer something like this, think this is the layer and this is the outer layer. Isn't it makes basal body for the flagella and cilia? Yes, it do. Okay, it do makes the basal body for the flagella and cilia. So there also, this is not what is that? Centrosome or centromere is not 100% needed. Empty OC is needed. That means something is needed to initiate the polymerization. Okay, some enzymes and proteins are needed for the initiation of the polymerization. Why I'm keeping in this only because still we don't know what are those all the exact proteins which are doing all the processes in there. Okay, still we are trying to find that only. Okay, what are the proteins, how they are acting, is a positive end protein, negative end protein, all those things. Still research is going on. Okay, that's why I also can't say complete. So still we are stuck only. So we'll try to find the way. Okay, I am also trying to go in that way only. Okay, so remember, think that this is the outer inner nuclear envelope, and think that this is the outer nuclear envelope. This outer nuclear envelope is joined with the our endoplasmic reticulum, especially our rough endoplasmic reticulum. Okay, that means it is continuous layer. Okay, this is not like completely separated and endoplasmic reticulum started. No, nothing like that. The outer nuclear membrane is directly started in smooth endoplasmic reticulum will also there, but in the most of the cases, the rough endoplasmic reticulum will be joined to the this chromatin materials. Okay, sometimes those endoplasmic reticulum will get rid of the ribosomes and they will form the smooth endoplasmic reticulum also. Okay, so what happens? That means this outer membrane layer is directly giving rise to the rough endoplasmic. Sorry, you can in simple way you imagine like endoplasmic reticulum. Okay, it will giving rise to the endoplasmic reticulum. Okay, outer and inner. So if you observe it in a clear way, you can see a smaller holes here, right? So these holes are, we call it as the nuclear pores, and also we call it as the pore complex. Okay, pore complex. Why we called it as a complex? We could have called it like simple pores, right? But if you observe it clearly, this is not a simple pore. This is a pore which is made up of the several protein structures. Okay, several protein structures which is joined again by the some other glycolipids and the glycoproteins. Okay, so what's the important thing then? Whatever the signal the DNA is sending, it may be in the form of RNA, right? Normally it may in the form of RNA, it's going through with this only. Okay, and whatever the signals which is coming from the cells from outside of the nuclear membrane are going fast through here only. Okay, and that's why it's highly selective. It will only allow the things which are necessary. Okay, do you really think you can directly go and meet Modi, or you can directly go and meet Yediyurappa? Right? It's not possible, right? A security will be there. Why we need a security? Because they are controlling the things. If something will happen to that, then it's a disaster, right? So that's why we are doing this. Okay, that's why we are doing this. How can you see a nuclear membrane then? Sorry, nuclear pores then? Nuclear pores can be visible under electron microscope. Okay, but the composition of the nuclear pores can be observed by different chemical procedures. Similar things what we did in plasma membrane, right? A similar kind of a thing. Okay, so these are highly selective. Nuclear pores are, remember, they are highly selective and they are made up of several bunch of the proteins, not a single, not a regular porine or not a regular channel protein. It's actually complex. Okay, it's a pore complex. It's made up of the several kinds of the protein. Okay, we'll move on then. So next one is a chromatin. Inside to that, what is present inside the nuclear membrane? Our chromatin is present. Okay, what is chromatin then? So in a simple way, I will put it in a simple way. Okay, So think that this is the DNA. Yes, this is the DNA. But DNA is several 100 meters long. Okay. You want to put it in a proper way. Okay. Then how you will put it? Then you have to like compress it, right? You have to condense it. To condensing it to somewhere, we need anything, right? We need at least a base to condense it. Yes, we have a base here, and you can roll this our DNA to this like this, right? As you can observe in here, right? All the flat structures, as you can see here, all of them are proteins. Okay, what kind of proteins these are? These are called as histone protein. Exactly, these are called histone protein. So what's the use of histone protein then? Histone protein give like the base, okay, base kind of the things. Okay, so Tejas, you deleted some of the messages. I didn't. Sorry for that. Okay, so histone protein means it's simply like if I hold this pen like this, and if I put some, what is it, a rope around this structure, then it will become the base, right? Exactly. These histone proteins will act as a base. Okay, act like a base. So when we first observed, we know that DNA is condensed in some form. Okay, but how we have to observe? Then we put some chemicals to that, or we can say dye. Okay, so I told you, right, we put the crystal violet dye for finding out is it gram positive or gram negative, or to finding out the this one xylem, we actually use the saffron. To finding out the phloem and all the things, we use the malachite green. Okay, exactly. For the chromatin, you have to use the different dye. Okay, that's acetocarmine. We normally use the nuclear dye. Okay, acetocarmine. That kind of several dyes are there. Okay, when you add a particular kind of a dye or fluorescent marker, then what happened? Certain region took darker color. Okay, certain region took the darker color. If you observe here, some of the regions, think that this is the dark color. Okay, here, where is the chromosome? We are watching inside the chromosome. We are not at all came up to the chromosome. We came from DNA. DNA is joined to form some structure only. Okay, we will come to the chromosome later on. Okay, some of the part took a dark color. Okay, some of the part, like this part here, okay, took light color. Okay, what may be the reason? Can you guys tell me? Some of the parts took dark color. I marked here. This part took the dark color, and some of the part took the very light color, like this part. Okay, what may be the reason? What may be the reason? Okay, exactly, exactly, exactly. So wherever dark protein is, dark color is there, there actually these chromatins, or what you can see, the DNA which is present towards the histone. Wow, that's what you call it as a chromatin, right? Chromatin. I will mention it. Okay, DNA with histone, DNA with sorry, condensed with histone, condensed form, right? This is like coiled in a very bit way, right? It's in a very tightly coiled. I can simply use the term tightly coiled. So there you can find the dark color because it's very tightly coiled or densely coiled. You can use the term also. Exactly. Where it is light, it's actually lightly coiled, lightly coiled. That means not that much of coiled here. Okay. So if you give both of this structure, okay, remember, don't think about it's a DNA, it's small, think that it's a big only. Which one is the tightly coiled dark, okay, tightly coiled structure, and for another person I will give you lightly coiled structure. Okay, which one do you think is easy to separate? Which one do you think is easy to separate? Which one? Answer me. Correct. Correct. It's a light colored or light coiled. Why? Because darker is tightly coiled. You need lot of energy to uncoil it, right? Uncoil it. But lightly colored is like easy to access. You can remove it very easily, right? That's why who have the lightly coiled will be the inner. Will be the winner, right? So that's what happens. Our chromosomes, chromatins, can be divided into two types. I am writing it here again. Okay, this chromatin can be divided into two types. The first one, we call it as the, one second guys, we call it as the euchromatin. Okay, We call it as the euchromatin. What's the meaning of euchromatin? Euchromatin means lightly stained, and this part is actually functional. That means these are the genes which actually code for your characters. Why? Because the enzyme can easily read this part. Okay, enzymes can go there. Probably you are observing here, right? RNA polymerase 1, RNA polymerase 1 enzyme which actually code or read the messages which is present in the DNA. Okay, so this enzyme can go and access that easily and give the message. Okay, functional form. And there is second form is also there, that is heterochromatin. So this heterochromatin part is darkly stained. Okay, it's darkly stained, and probably you guys are already guessed it. The enzyme can't go and read it out because it's too complex. Okay, it can't go and read in between. Just imagine it don't have the space to go in also, right? So these are non-functional. That's why we sometimes call it as waste also, or junk. Okay, non-functional. That's why we also call it as the junk genes or junk DNA. Okay, junk DNA. Junk DNA means what? These are not at all coding by the any enzymes. Okay, these DNA is not at all coded by any enzymes. That's why those characters are not at all possible or visible. Probably some of you may have a superhuman character. Okay, superhuman character. Probably you have the character to fly like a Superman. Okay, but probably that particular gene is present in darkly stained structure, or it may be in a heterochromatin structure. The enzymes can't go in read in there. That's why you can't fly. Okay, so that's what happened. We don't know. Probably you may have or not, right? So heterochromatin is non-functional or junk DNA. Probably it may had some function one time it may had some function, but as for our knowledge for now, it don't have any function. Okay, I will repeat it one more time from the initial. Listen, the DNA will coil it around the histone protein and form a condensed structure. That's what we call it as the chromatin or chromatin thread. Okay, you can use the term thread also, no problem. Okay, thread. What happened? This chromatin thread which is present inside our nucleus. Okay, what's the red colored part? It's the enzyme. Red colored part is the enzyme. Okay, so what happened? The chromatin may be present in two forms. One is the tightly coiled form, or another is the lightly coiled form. Tightly coiled form take more strain, and this is not at all readable, non-functional. That's why we call it as the junk DNA. Okay, or heterochromatin. But there are some parts, these are not that much coiled, or you can say lightly coiled. These can be accessed by the RNA polymerase or enzymes. So this can be readable. That's why we call it as the functional chromatin or euchromatin. Eu means true, right? True chromatin, true genes. Okay, this is related to chromatin. Next, we came to the nucleolus. Okay, nucleolus. Here you can observe two structures here or two diagrams here, right? In first diagram, as you can see here, this is the nuclear envelope within the nuclear pores or nuclear pore complex. Okay, inside to that we removed the DNA. Okay, DNA is removed or chromatin is removed, only left with a dark structure or the nucleolus. Here is also you can observe. So here is the cytoplasm. Okay, here is the nucleus. This is the complete nucleus, a large nucleus you can think. Okay, this is the nucleus. Inside the nucleus, a dark structure is present. This is nucleolus. Okay, this is the nucleolus. Then what is nucleolus then? Is it the center of nucleus? No, not like that. Nucleolus is aggregation of enzymes and proteins. Enzymes and protein. This is just the aggregation. Remember, it do not have any layer, membrane or nothing. Okay, it's just the aggregation of proteins and enzymes. Okay, what is the function of then nucleolus? Okay, the function only matters. Okay, that's what matters, right? The function is production of rRNA. there is a type of RNA which is called as rRNA, because probably you guys guessed it, right? Because rRNA present in the ribosome. R refers to the ribosome. The RNA which is present in the ribosome, you call it as the rRNA. So production of rRNA will happen here. Okay. What is the other function then? You can continue this. Okay. Assemblage or assembling or the of ribosomes. That means, you know what is ribosome, right? What is ribosome? Ribosome is the aggregation of the RNA and the proteins, right? How the RNA is integrated inside the ribosome. If I write a diagram something like this, then I will write RNA something like this, right? Something like this. There are like two types of RNA normally. Then how this RNA like present inside that protein, how it is arranged in that manner, that assembly or that arrangement will be done by this particular nucleolus. Okay, this is the importance related to nucleolus. That much only, not anything special. What are the enzymes or proteins? Those are not needed. Okay, those are completely different kinds of enzymes and proteins will be there. Use it for the conversion of DNA to RNA, RNA to protein, that kind of the job. Okay, then we will move on to the chromosomes. So you know about the chromatin. What is chromatin? It's a condensed form, you know. Okay, what happens if you super condense the chromatin? Okay, so then you will get the chromosome. Chromosome is the highly condensed DNA. Okay, highly condensed DNA, you can call it as the chromosome. Okay, so how it will condense then? Do you really think it always condense in this form? Exactly. And when it will condense? So condensation happen. Anybody know when condensation will happen? Condensation will happen during cell cycle. When a particular thing or particular cell want to go for the this one, not in a S phase actually, in the initiation of prophase. Okay, when it's going to the cell cycle, then the condensation start. Okay, not in an interphase, normally in the prophase. Okay, so why it have to go through that? Okay, why it have to go through that? That's actually questions, right? See, again, think about your earphones. Okay, think about your earphones. Think that like four friends, like any four of you are went to some picnic or something, and you took only one bag and you put all of your earphones in a single pocket. Okay, you traveled 1 day or 2 days, leave it. Okay, after 2 days you want your earphones. Okay, when you took that back, everything is like shuffled and mingled, something like this. Okay, something like this. Okay, all of you like something jumbled like this. Then you have two options. What you can do, you can four of you hold your earphones and pull it together at the same time. Okay, otherwise someone have to sit and have to untie all the knots and everything, have to remove that. Okay, both of them are problematic. Why? If you do the first thing, if you pull in like four corners and if you pull it, what happens? Everything will get damaged, right? Everything will be, every piece will cut down. Then you can't, none of you can't use that, right? And if you go for the second step, someone will sit and untold all these things. Then what's the problem then? It takes a lot of time, right? It's like unnecessary time. Nobody wants to do that, right? So much time it will take. Then what's the good thing? Before you going for the trip, you can arrange it somewhat like this, right? You can arrange it somewhat like this, and you can put a tie here, right? So I will write the another one earphone also. So you can put something like this, and you can put tie here. Think that this is the earphone, right? If you put like this, not 2 days, if you go for 2 months of traveling, after also you are taking, nothing will happen, right? Because everything is separate. You tied with this, you put some rubber or something, right? Everything is already how can I say, it's there, you can directly take it out. It's time saving and it can ease the process, no damage will occur, right? So much easy, right? That is what the use of the chromosome. Okay, if it is in a chromatin state or in a DNA state, it want to got separated. Remember, you don't have a single chromosome like prokaryote. Okay, so you have how much chromosome? You have like 26 chromosomes, okay, 26 chromosomes, and everything will mingle together and like everything will become piece, okay, like Ravichandran, it's piece. Then you can't use any one of that, okay. So they should arrange properly, exactly, Manvita. It should arrange properly. That's why they are going for the condensation stage, okay. They are going for the condensation stage, okay. So if you imagine this particular only, if you take this particular chromosome only, how they arranged, the DNA molecules will be something like this. DNA molecule will be something like this, or histone protein arranged things. So this looks almost like this particular structure, okay. It's somewhat like this. Everything is joined together to the histone protein, right? So it's somewhat like this. So then it can decondense whenever it wants, right? Yeah, something like it's like rolling, right? As you can see here. So in between that, you can find the histone protein in between. So like that, okay. So here is one of the image. Here is another one. It's not condensed also. Each divided cell give it DNA only. DNA only, but not similar, right? If you pull together 4 earphones, then there is how much probability you get, you will get all the pieces of that particular earphone, right? It will be cut down, it will be cut down. You may get one gene, you may not get other 5 genes. That's what happens, right? That's why they are going for this one, okay. They are going for this one. So they normally go when there is a during cell cycle only, okay, not other than the cell cycle. Then during the cell cycle in interface only it's get divided, so it looks like this. So we will see, okay, how the shape in different in this cell cycle chapter. No need to take tension, okay. Condensation will happen during the cell cycle. That's why it looks like this, okay. So each of that you can call it the one of the arms, okay, arm. You can call each one as the arm of that, okay, you can call it as the arm. Kai, hands, okay. You can call it as the hands. And there are constrictions are there, okay. What's the meaning of constriction? So if I write a chromosome like this, there should be something where you put the rubber band, right, in that particular earphone. That rubber band is the constriction. So it is not like something like this. It is not something like this, right? It's our constriction like this. This constriction, you can call this as this constriction is called as the primary constriction. Primary constriction. Or you can also use another one term. What is that another term? You can also use another term. There is the centromer. Don't confuse with the centrosome. Okay. Don't confuse with the centrosome. Okay. It's the centromer. Okay. Yeah. Okay. So that's different and this is different. This primary constriction. Remember constriction. What is happening, right? Constriction. So that is what we call it as the centromere. There is no other special structure is there. Okay, there is no other structure. There are some proteins will be there. That's it. On this constriction, there are some proteins will be present on 2 sides. Okay, on this, so there are some proteins are present. I will use the different color, no issue. A yellow color I will use. Okay, this protein only on 2 sides. Remember, not completely surrounded. Only on 2 sides are present. This is called as kinetochore. Kinetochore protein, or you can say kinetochore. What's the use of the centromere or the primary constriction? The function is it is holding the chromosome, holding this two how can I say sister chromatids, holding the chromatids. Okay, so I will tell what's the chromatids. Okay. Correct. Very good question, Akshay. Very good question. Okay. Remember, whenever you are considering this, always consider this is the one arm, this is the one arm, and this is the another arm. Okay. But whenever considering the gene, don't cut it like in this way. Okay. Always cut it horizontally. Okay. If you cut it horizontally, you can find this one, right? This is called chromatid, one single chromatid. And this is the another single chromatid, and both of that joining we call it as a sister chromatid. Okay, why we call it like a sister chromatid? Because both of them having the same origin. Okay, both of them have the same origin. One will give rise to another one. That means they are similar in genes, structures, and everything. That's why we call it as the sister chromatids. there should be something to keep the sister chromatids joined, right? Sister chromatid to joined. That's called primary constriction or centromere. Okay, exactly. That may be different, Prajna. A centromere should not be in middle only. It may be above, below, everywhere. Okay. So, what is the function of kinetochore then? The function of kinetochore is attaching of spindle fiber, attachment of spindle fiber. Kinetochore will give the site for the attachment of the spindle fiber. Site for the attachment of the spindle fiber. Okay, this is relating to chromosome. Then you can see here, as Prajna asked, is centromere should be in the center? No, there is no rules like that. Okay, there is no rules. Then how it will be then? Okay, just look here. The centrosomes are sometimes present in the middle. Okay, present in the middle. Good question, Manvita. So before the replication, that means before the S phase, our chromosomes will look something like, there is like no condensation. If you do the condensation, it will look like this, okay, with a single chromatid. Okay, yeah. So some people are write it down. Okay, remember guys, whenever you want to cut the this sister chromatids, cut horizontally, never cut it transversely. Okay, always cut it horizontally. Okay, remember this. Okay, if you do condensation before S phase, okay, in the interface, who know about this only think about it, who don't know what is S phase, not at all a issue. We will see that in the replication. Okay, vertically, sir. Correct. So it will looks like this. Never cut it vertically. Like this. Never cut in this way, not in this manner. Okay, you have to always cut in this manner. Okay, horizontal manner. Vertically means what? Vertical. Correct. Like this. Vertically. In this manner. Okay, from here to here, top to bottom. Should not correct. From top to bottom. Okay, exactly. From top to bottom. You have to cut from top to bottom, not in other way around. Okay, from here to here, don't cut. From top to bottom, you have to cut it. Okay. So thank you guys. Okay. So here what you can see here. So centromere is here, and this chromatids, like both the arms, okay, upper arm and lower arm, both are having the exact same size. Okay, same size, right? Both of them are in the same size. Then we call it as the metacentric. Okay, metacentric means what? The centromere which is present in the middle. Okay, same size, and centromere is present in middle. It is present in the middle. Okay, but not always like this. Sometimes one of the arm will be smaller, as you can observe here. One is smaller and another one is the larger. And this is centromere is not middle, right? Not in middle, or you can say little away from the middle. Little away from middle. So then we call it as the submetacentric. I'm just writing it again because probably some of you can't read that words, right? That's why. Submetacentric. Okay. Sometimes this is almost in the tip. That means only one side, a small arm will be there. Other side, a long arm will be there. That means the arm is in the head, right? Arm is look like a head. That's what we call it as the acrocentric. That means almost in the head. Okay. Last, one side there is no arms will be there. There will be not at all a single arm, and other side one arm will be there. That means only one arm. So that's what we call it as the telocentric. Remember, telocentric chromosomes are not present in human. Okay, not in human. But other things can be present in human. Other chromosomes are present in the human. Okay. No, not at all. None of them are haploid. We can't say haploid, but only one chromosome is present. I told you, right? How can you tell the chromosome number? You can tell the chromosome number based on the centromere. If you divide it something like this, then it's only one centromere is present. That means haploid. All of them are haploid only, right? Don't consider based on the arms. Haploid, diploid based on the centromere. Always remember that. Okay. Why cannot? Don't know, Srishti. Like why we don't know the telocentric is not present? Don't know, right? These are the things which is present. So it is not present in here. Okay. Metacentric not present in some other organism, right? That happens always. Okay. Probably due to the disjunction or something, it lost. Probably we never formed this. Who knows, right? How did they got photos of the 20? I will tell you that one. Okay, one second. So as you can observe here is the nucleus. These are the chromosomes. If you go zoom the chromosome, you will get the DNA molecules arrangement, right? So remember about the different types also. Okay, all these are haploid or diploid. All these are haploid only because I wrote only one chromosome. If I wrote something like this, then I can call it as the diploid, right? Two are there. By I wrote only one, so it's haploid only. Okay, remember guys here. So the smaller arm is called P arm. Okay, P arm. What is the meaning of P arm? P arm means a smaller arm. Okay, wherever you encounter a particular metacentric chromosome, one arm is smaller and another one is larger. Then the smaller arm we call it as the P arm, and another one, that means long arm, we call it as the Q arm. Okay. What about metacentric chromosome then? What about the metacentric chromosome then? In metacentric chromosome, both arm will be normally same length. Okay. That's why don't consider this here. Okay. But normally we will consider upper arm or arrangement in alignment, which is present normally upper towards a P arm, or some people also consider both as the P or Q arm. Okay. So that's there. But normally the shorter arm is P arm and longer arm is the Q arm. Okay. So here you can see some of the microscopic images. Okay, even in the light microscope also you can observe the chromosomes, not at all an issue. And this one is actually human this one. Okay, human chromosome structures. Okay, they used with the fluorescence microscopy. Fluorescent microscopy means we add some of the dyes, that means fluorescent dyes that will attach to the some of the parts. Okay, here you can observe this one is the metacentric chromosome. Here is the centrosome, centromere. Okay, this is the metacentric chromosome. If you observe, these are these things are, or if you go for some here, okay, these are submetacentric chromosome. One is longer, one is shorter. Okay, and if you observe, these are 15th pair, or this one 14th pair. These are acrocentric chromosome. Look, one is longer, another one arm is way too smaller, right? If you are in zoom, you can like zoom little bit. Okay, if you are in zoom, you can zoom. Okay, why they are not attached? It's not attached. We take a separate photos and we arranged it in a manner. Okay, as per the our like we gave the name depending upon their size, which is called as karyotyping. Okay, we call this technique as the karyotyping. Okay, I can't write it properly here. So karyotyping, CARIYTYPING, karyotyping. Okay, we collected the photographs and we arranged it based on the size. And here you can see X and Y. Look, Y how much smaller it is, X how much smaller it is. Okay, that's why they are not attached. Okay, 16th one is human DNA or color. No, we use different colors. Okay, 16th chromosome. 16th chromosome is here. They left a little gaps because they are arranged it in a based on some structures. Okay, and then no, this is not haploid. Remember, guys, all these are diploid. You can see pair, right? 1, 2 contains 2, 3 contains 2. This is all 46, right? It's a diploid one, right? If you observe. So we took a photos of alignment in the metaphase stage. Metaphase stage only, because there only is highly condensed. We took a photos, then we cut the photos and we arranged. That's how we will do the karyotyping. Okay, so whenever we are doing the now there is softwares are there, that is secondary, but whenever doing, we are cut those things and we are arranged. No, I told you, right? We just took a photo, then we cut the things. We added only the this ones, fluorescent dyes which are attached to DNA. Okay, that's why they are only attaching to DNA and only the only chromosomes are visible to you guys. If you use it for fluorescent dye for a protein, it will attach to some more proteins. We can't see the chromosome then. Those are highly specific. Okay. Yeah. So in this particular one, here is also you can see only the you can see chromosome, not about the threads or anything, because threads are too tiny to see. Using fluorescent dyes only, we can see that. I will show that photos also in the next class. Okay. If there is one chromosome less than body, then what happens? If that chromosome is first, second, third, or some other chromosome, any chromosome probably, apart from sex chromosome, any other chromosomes, if you lack one chromosome, either it will cause the complete, how can I say, disarrangement of your body, or most probably it will be aborted without the notice of mother only. Okay, without the notice of mother. So it know that all these chromosomes will give certain character in the development of body. If you lose something, it won't develop. Okay, that body won't develop at all. Okay, but addition will something happen. We will see that in the second year. Okay, sometimes some addition will happen or deletion of some part will happen. Then what happens? The person will be there. He may lose some things. Probably he may lost good intelligence, or he may lost sensing of the pain. Okay, or sometimes he may lost ability, or some organs will be less developed. Okay, all those things will happen. So these are also photo of a chromosome, just a random photograph, not about anything. Okay. So one question came. The question is simple. Okay. In mitosis, spindle will join to chromatin, centromere, kinetochore, or DNA. I'm just launching the poll. You can answer the yes, you can answer in the poll, and who are in the YouTube live can answer in the YouTube live chat. Okay, I'm getting a lot of answers. Nobody leave the class, okay, because we have very important information to tell you guys. So don't leave the class before I tell you to. Okay, Darshan, your train started. Last 20 seconds, guys. Hi, Anum. You came now or what? I didn't see. No, Chiranta. Like you misunderstood. I will explain. Okay, I will explain that. Don't think like that. I will tell you how it will be different. Okay, then I'm ending the poll. It's already more than 1 minute. Okay, I'm sharing the results. 4% chosen option A, 47% B, 64% C, 4% D. Exactly. Remember, here only I will explain the things what is bothering you guys. Okay. Mitosis, spindle, they are asking, leave those things. Okay. Remember, if I write, if I take regular this one's cell in your body and condense the DNA, I will get the DNA like this. Okay, get the DNA like this. So this chromosome is from your father, and this chromosome is from your mother. So here you can find one centromere, and here one more centromere. That means constriction. So you can call this as the, how can you call, diploid, right? Diploid. That means 2n. 2n, that means 2 chromosomes are present. Okay. After the S phase, when division is happening, this gives rise to another one set. That means this only replicates, nothing will changes will happen. Only this will replicate like this. Okay. So then what is this? These are diploid or tetraploid. Still these are diploid only. I told you to remember the chromosome number based on the centromere, right? And what about these things then? These two things are originated in similarly, right? So we call it as the sister chromatids. That means what is the meaning of chromatid? Chromatid means a single type of the chromosome, something like this in a chromosome. Okay. Then how you call these two things? You call these two things as sister chromatids. How can you call these two things? If you join both of these, you can call this as the homologous chromosome. Homologous chromosomes. Homologous chromosomes means what? They have a different origin. That means one from the father, another one from the mother, but have this similar quantitatively. They are same. That means have the same size. This is also first chromosome only. This is also first chromosome only. Remember, what is haploid then? Haploid means simply like this. Okay. If even in the there is no pairing is there. There is no pairing is present. Only one chromosome is present. That is what you call it as the haploid. Okay, haploid, or you can call it as the N, right? So if in your regular body cells, you can find 23 these kind of cells from the father cells, 23 these kind of chromosomes, 23 father chromosome, 23 mother chromosome. But in sperm, if I take a sperm, only 23 are present, not 46. That is haploid. But your regular body cells, these things are present. Body cells, that is diploid. Body cells, that is diploid. Okay. What is the difference between kinetochore and centromere then? Centromere is just a constriction. Chirant. After S phase, haploid also go for the division, okay, replication. Then how it will look? It will look something like this. Still, it has a single chromosome, sorry, single centromere, right? Single constriction only, right? So still, it's a haploid only. That's why. We will see that one more time. Exactly. Centromere means a simple constriction. Simple constriction. Okay, nothing more. Simply like this. Simply like this. Okay, just a structure. Exactly. What is kinetochore then? Kinetochore are the proteins which is present on the centromere. Okay, proteins which is present on the centromeres. If I take this particular chromosome, how many kinetochore are present? 2 kinetochore are present. In this 2 kinetochore are present. Okay, how many centromeres? Only one centromere here, only one centromere here. Remember, centromere is only one. I will writing it one more time in a zoom. Okay, so think that these are the chromosomes. Okay, these are the chromosomes. This is the centromere. In between, a condensation is happening, joining. But kinetochore means the protein which is present here, both the sides. Okay, something like this. Remember about this. Okay, so this our what is that? Microtubule and comment attached to kinetochore. They never attached to centromere. It's given wrong in the textbook, I think. New textbook updated, I think. I'm not sure about it. But normally, most of the people will say that attached to centromere, but it's the wrong statement. The microtubules or the spindle fibers or asters will always attach to kinetochore, not to the centromere. Okay. Yeah, that's what happened. So the correct answer is kinetochore. Okay. Then what's the information I thought of giving you? Sorry, I left with this one thing. Last one we left with the microbodies. Microbodies are simple structures with a single lipid membrane. Okay. Microbodies are things with these single lipid membranes. Okay. These may contain spirosomes or peroxisomes. Okay, peroxisomes. So different things. Okay, majorly these two are important. This only contain a lipid layer, and inside there are some solution. Okay, if you take a peroxisomes, this normally contain hydrogen peroxide. Spirosomes normally contain some of the this one storage materials or some of the enzymes. Okay, so how these bodies are helpful? These microbodies normally formed by the lipid bilayers only, lipids only, which is actually produced from the regular endoplasmic reticulum. But these are different from the plasma membrane because they don't have any protein or any other structure. Okay, they don't have protein or any other structures. These are simple, a ball-like of thing, or you can think about balloon. Inside of that, important enzymes or the liquids will be present. Okay, so we will learn more about peroxisomes and spirosomes later, but this much only given, not anything special in that. They don't have DNA, they don't have ribosome, or they don't have anything else. Okay, plasma membrane, where? Oh, sorry. Okay, so they given plasma membrane. Normally we call it as lipid bilayer. We normally won't call it as the plasma membrane. Okay, so sorry for that. We normally call it as lipid bilayer. This structure is not exactly like our cellular plasma membrane. because they don't contain all the porins or the integral kind of the proteins and all those things. Okay. And the difference, last one, the difference between plant and animal cells. So probably you may already guessed it. Plants will have the cell wall, animal don't. Plastids are present here, it's absent. Large vacuoles will be present here. Normally vacuoles are not present, even though it will be very tiny in the initial phases. And centrioles are absent here, and it will be present in here. Okay. So that much related to that. The important thing I want to tell you is this week you have the test. Okay. So B4 chapter test will be there on this Sunday. Okay, that's the important thing I wanted to mention. Okay, and just launch the NPS. You guys will be like started to write the NPS, attend the NPS, then we can discuss this. Okay, so remember, who are in the WhatsApp know how to attend all the things, and who are in the YouTube or in the Telegram group, we will send the PDF in the form PDF in the Telegram and WhatsApp group, Telegram group, and also the WhatsApp group. And a Google form will be given. Google form, you have to attend the answers or fill the option in Google form. In Google form. Okay, the timing will be informed. Timing will be informed. Will be informed. Okay, so yeah, for both of the things, for Telegram people also, and also the people which are present in the WhatsApp also. Remember, this is super important. Okay, so yeah, last one. How can animals give birth to the more young ones at the same time, but why can't humans? So it always depends. Okay, this week three subject test. Okay, yeah, be ready for that. Can we leave the meet? Once you completed the NPS, you can leave the meet. Okay, no problem. So remember, so always giving birth to young ones depending upon the nature. Okay, normally based on their environment. Okay, so what happens for the lower organisms? There are less security around them, right? For the newborns and everything. That's why they have to create more and more progeny to continue their generation. For humans or the higher organisms, they don't have that problem because we can secure our young ones. So even a single child is enough. That's how it's always go on, right? So depending upon the environment, how it's pushing the things, it will happen. Centrioles are absent. Correct. Centrioles are absent. That's what we told, right? Centrioles or centrosomes, both are same. Centrioles or centrosomes. Not centromere. Centrioles or centrosomes are absent. Yes. Anything else, guys? Any more doubts regarding this chapter? No. Like simple. DNA like this. When DNA is joined with the histone protein, it forms like this. When it supercoils, it will make something like this structure. This structure is the chromosome. Can you understand now? Like these are the histone protein. So this is the DNA. This is the chromatin. And this is the chromosome. So what is happening along this line? Condensation increases. Condensation increasing. Increased condensation gives rise to increased different kinds of things. Okay. A pore complex normally present in both of the things. Pore complex is present in nuclear membrane structure, right? Nuclear membrane structure. It is present in both the things. Yeah, also in plants and also in the animals. Chromosomes is highly condensed DNA. Exactly. Chromosome, you can say highly condensed DNA or highly condensed chromatin. Yep. Bro, did you understand or you still have the doubt? It should be, right? All chromosome, whatever the chromatin or DNA present in your cell, will form the chromosome only. When it's going for the cell division, it always go for the chromosome only. It can't present like loose function, right? Because it will be cut probably. So all chromatin will form the chromosome. So this is the different phases in a cell cycle. DNA or chromatin phase is like regular phase. Chromosome phase only happen when there is cell division. Okay. King, like bacteria we already saw. Only issue is like fungi, right? Fungi is almost like the plant cell only. The difference is like instead of having this cell wall, cellulose cell wall, it have a chitinous cell wall, right? And it also, and one thing is it don't have the this one. What is that? How it's different? This one, chloroplast. Okay, it don't have the chloroplast, and it also don't have the centrosome. Okay, fungi also don't have the centrosome. That's why in the initial we kept it in the plants only. Pore complex which is present in the nuclear membrane. Plasmodesmata which is present on the cellular membrane. Think something like this. This is one cell. This is another cell. Okay, and this is one nuclear membrane. This is another nuclear membrane. Okay, so the pores which is present here is the nuclear pores. Nuclear pore. Okay, the pore which is present in between two cells is plasmodesmata. Okay. Just seasonal birds. Most probably, bro. I'm not sure about it. Okay, I'm not sure about cheetah. But no, most of the cat species are seasonal breeders only. By considering that, probably cheetah is also seasonal. But I'm not sure about it. The issue is like every organism will be different in their patterns, right? So if I know only, then I can answer that. Any more doubts, guys, relating to this chapter? So I already sent the modules once. So I will send the module in Telegram today. Okay. Please try to solve the more of questions. Okay. Okay, guys, then I will end the session. I will end the poll first, then I will end the session. Okay. Okay, I'm ending. Bye-bye.